Hello, everyone, and welcome to today's live broadcast. Rapid whole genome sequencing in the NICU and PICU decreases infant morbidity and mortality. Presented by Stephen Kingsmore, President and CEO, Rady Children's Institute for Genomic Medicine. I'm Alexis Cross of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your question into the drop-down box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Kingsmore. I will now turn the presentation over to him.
Good afternoon, everybody. Welcome to my office here in sunny San Diego. So this afternoon, I'm going to talk about rapid whole genome sequencing in the NICU and PICU, and hopefully show you how we are decreasing morbidity and mortality for our youngest and most fragile patients. I have no disclosures. We're going to start with a case report. This dates back to October of last year, and this is family 243 enrolled in one of our research studies. The little boy is called Maverick, and he was admitted to our neonatal intensive care unit from the emergency department at 8 days of age. He was presenting complaint. He had status epilepticus. That means unremitting seizures, so seizure after seizure after seizure. The background is that his mum is 23 years old, and she's previously had one child. The pregnancy was uncomplicated, with the exception that ultrasound picked up a slight increase in the size of Maverick's ventricles. He was delivered at an outside hospital by C-section and was doing well immediately afterwards, and discharged home on day of life 3. So doing well until 8 days of age, when he started to seize, resulting in his admission. So a case like this is a medical emergency, and we started to evaluate what was the cause of his seizures in order to give him the appropriate treatment. One of the first tests is an electroencephalogram, which is shown on the right-hand side, and that showed two things. First of all, the spiky activity is actually seizure activity. Second thing is that the background profile, which should be wavy, instead was very flattened. We call this burst suppression. This combination of seizure activity and burst suppression is rather ominous and indicates a very severe and potentially lethal form of neonatal seizure disorder. As part of his workup, he was given an intravenous dose of pyridoxine, vitamin B6, and there was no change in his EEG as a result of that, suggesting that he did not have vitamin B6 responsive epilepsy. He had a brain CT scan, which was negative in terms of illuminating why he was seizing, so no evidence of intraventricular hemorrhage or acute birth injury. He was worked up for infectious diseases, another common cause of seizures in infancy. That was negative. And he had a lumbar puncture performed, which showed that his lactic acid level in cerebrospinal fluid was grossly elevated at 6.3. This indicates that he was really critically ill and that these seizures were resulting in brain damage. And finally, his serum CK was also grossly elevated, although not to the extent that it could injure his kidneys. So over the next 12 hours, his disease progressed. This is a note from his medical record. Last night was rough with ongoing seizures, continued despite the 2 first-line treatments. By the following morning, the seizures were not any better. He'd now worked his way through our entire list of drugs for anti-epileptic drugs, and he was on a midazolam drip, that's a benzodiazepam, and the dose of that is increased until he stopped seizing. Unfortunately, in his case, he continued to seize, and the drip was increased until he needed to be intubated for respiratory failure. At this juncture, our pediatric neurologist, Dr. Gold, started to discuss the prognosis with his mom and dad and let them know that this particular form of neonatal encephalopathy is associated with a bad prognosis, just to prepare them for what the future might hold. So we have several questions at this point in this little baby's case. What's your top differential diagnosis? Obviously, you can't tell me, but be thinking about that. What do you think is the top differential? What's the next test that you'd order in Maverick's case? And then why would you order that? And how might it be impactful? This particular baby at Rady Children's Hospital, the next test ordered was rapid whole genome sequencing. And we're going to go over in the next couple of slides the answers to these questions. So first of all, looking at babies in the NICU or in the pediatric intensive care unit, the PICU, this slide summarizes kind of the state of the art in terms of management of these cases. So about 15% of babies or infants will be admitted to a NICU or a PICU in that first year of life. They'll immediately receive empirical treatment based on the top suspicion of the physician. They'll get better or worse going around the circle. In response to that, they'll have modifications to their treatment, and all the while there'll be a search for the underpinning etiology underlying the reason that they are in the NICU, in this case, seizures. On average, it takes about 60 days to reach a diagnosis using traditional genetic tests. Clearly, in the case of Maverick, many babies, infants in the NICU and PICU, that's far too long to wait. And so we've developed rapid whole genome sequencing to circumvent this. With a 60-day turnaround, babies frequently have been discharged home or transferred to palliative care, or indeed have died prior to a diagnosis being made. Precision medicine is really vital for babies with seizure disorder. On the right-hand panel there are listed a number of genes that cause seizures in babies and a specific treatment regimen, which is not the standard test treatments that we would give to babies with seizures, is listed on the right. And so in the absence of knowing what causes a baby's seizure, we don't really know which treatment specifically to give. And in fact, amongst the 8,000 known genetic diseases, there are over 850 that cause seizures. And so just guessing which seizure disorder this baby might have is almost universally going to result in a wrong guess, and thereby likely a wrong treatment regimen. In fact, these diseases are the leading cause of death, genetic diseases in the NICU. So this is not a small problem around the country. About 370 inborn errors of metabolism, for example, so metabolic conditions or biochemical conditions, are associated with seizures, and 90 of those have effective treatments. So if we diagnose it promptly enough, we can give an effective treatment and prevent the baby from having any further neurologic impairment. And so over the last several years, we've developed methods to make a diagnosis by rapid whole genome sequencing, or RWGS, in about 2 days, which allows us to provide rapid precision medicine and improve outcomes. So let's just walk through that together in this particular case. So the first thing to do is for mom and dad to give consent for their baby's genome to be decoded. Maverick's mom and dad gave us that. Second, we take a small blood sample, which we race across to our genome center, which is located on the other side of the campus from our neonatal intensive care unit. There, one of our certified laboratory specialists, such as Luca, shown here, prepares that DNA for sequencing. That's a step called library preparation, and it takes about 2.5 hours. Next, we load that library, which contains the genome sequence, onto an Illumina genome sequencing instrument. And here Sarah's shown loading one such instrument in our facility. We actually used this technology to recently set the Guinness World Record for fastest genetic diagnosis in a baby. So it takes about 15.5 hours for us to decode the genome 45 times. Next comes the computational phase, where we go from that 125 billion letters of DNA sequence. That's the equivalent of about 45 Empire State Buildings if we were to actually type this out on pages, 8.5 by 11 pages. It would be 45 Empire State Building level loops. Quite staggering. So that's 125 billion letters of DNA sequence. And using technology from Decode Genome, in about 30 minutes, we take that to 2.8 billion letters of, in this case, Maverick's individual genome sequence being assigned. And then secondly, to identify the 4.9 million letters where Maverick's genome is different from the reference genome sequence. So it's in this space that we're looking for the cause of his illness. The next thing we do is to go back to his medical record, which is shown on the right in the slide. We use a different technology from a different company called CliniThink to perform natural language processing. That means that we're using machines to read the medical record and identify all of the clinical features that characterize Maverick's illness. And what you can see here highlighted is the word seizures. And not only does this program pick up all of those features, but it also translates them into machine-readable terms called human phenotype ontology terms, which allows us to automate making a diagnosis. This step takes about 20 seconds. Here we see Maverick's features, all of the features of his disease in a couple of different colors. The red circles are the actual features that we observed in his medical record. Blue features are what you'd expect to see in his disease according to one of our textbooks called Online Mendelian Inheritance in Man. And then in gray, dark gray, two features where you see an overlap between the textbook description of his disease and what we're actually observing in his medical record. And this just underscores that in babies, one of the reasons it's so difficult to make a diagnosis is because the features of their illness look so little like the textbook description. Basically, there's a lot going on in a newborn baby, some of it related to the genetic disease, some of the fact that he's a newborn, some of it relating to the fact that he's in an intensive care unit. And so this really confounds the ability to make a diagnosis based on observation and clinical expertise. When we looked at 76 babies with genetic diseases, we were able to measure just how divergent they were from the textbook. So the blue box here on the right is the textbook description of the average number of features in each child and their genetic disease. The red circle shows what we observe in the medical record of those children. And you can see the overlap's only 3 features out of about 93. Amazing how little concordance there is. And then if we use just human reading of the medical record to identify what we perceive to be the key features, on average, we only extract 5, and they have very little overlap with the textbook, only 0.9. And so machines can do the job better than humans and give us a more accurate picture and a more comprehensive picture. So the next step in analysis involves the medical record. The first thing we do is to remove all of the variants in Maverick's genome, which are common. Maverick has an unusual condition. It cannot be caused by a common variant in a genome. So that gets us to about 700,000 variants that are left. Next, we identify the letter changes that could cause disease. That's those that, based on a whole bunch of prediction algorithms and database lookups, we think these variants might cause disease. And in Maverick, that number is 962. And that's true of everybody who's watching. Each of us in our genome have about 5 million DNA variants, about 700,000 that are uncommon, and about 1,000 that could potentially cause disease. We then marry that 962 with the clinical features that we observe in Maverick, 159, and then with almost 5,000, or just over 5,000, genetic diseases, including all of the causes of seizures in babies. And when we look at that intersection of 962 by 159 by 5182, we're left with a single variant that matches his clinical features and matches a genetic disease. And that condition is our diagnosis. And here is actually a screenshot of the software algorithm with that result. And this has occurred about 20 hours after we started the testing. And in this case, Maverick has 2 changes in his genome, a C changing to a G, it's a cytosine, to a guanosine, and a guanosine changing to an arginine. They're both in the same gene. It's aldehyde dehydrogenase, type 7, subtype A1, or ALDH7A1. We have the chromosome position, it's on chromosome 5 at the 2 positions shown. And one of these, the top one, is a missense. It changes a glutamic acid to a glutamine at amino acid 427 of the ALDH7A1 protein, and the other causes a premature stop in the protein, making a truncated protein that's 110 amino acids long. One of these is inherited from mom, one from dad, and so mom and dad are carriers. Maverick is compound heterozygous for 2 variants, giving him a diagnosis of pyridoxine-dependent epilepsy, with 1 copy inherited from his mom and his dad. Now, what's remarkable about that is that this is a treatable condition. And here we see in this slide on the bottom right, ALDH7A1. This is the protein which is broken in Maverick's brain. And this is a biochemical pathway showing what that particular enzyme does. It's involved in the degradation of L-lysine, shown at the top right. And so this is a series of biochemical steps that degrade dietary lysine so that it can be eliminated in urine or in bile secretions. In Maverick's case, because this protein is broken with the X there, he has pyridoxine-dependent epilepsy. And that means that he has a buildup of the precursor that's supposed to get broken down by that protein. And you can see the name for it there, amino adipic semialdehyde, or AASA. So this is building up in Maverick's brain. It spontaneously converts to the other red circle with P6C. Now, both of these compounds are neurotoxic and partly responsible for Maverick's symptoms. But the other thing is that P6C, shown on the bottom right, spontaneously complexes in the brain with vitamin B6, or pyridoxal 5-phosphate. meaning that there's no free vitamin B6 to participate in 25 vital biochemical reactions in his brain. And that's the main reason why Maverick is having severe seizures. So as I said, this is treatable. Number one, we restrict dietary lysine so that he's not making much of these toxic precursors. And second of all, we give him a lot of supplementary vitamin B6 to overcome the fact that it's being complexed. And so 39 hours after enrollment, our lab directors, Shamol and Sharif, shown here, call up the neonatologist who's on call. It's about 10:30 at night with the result and the advice to give Maverick the treatment regimen we just discussed. And so 55 hours after consent, that treatment started, triple therapy with pyridoxine or vitamin B6 injections, restriction of lysine, and supplementation of arginine. And this combination overcomes in large measure the biochemical problem that he has. There's a really nice response. His EEG, which I showed you in one of the first slides, goes back to a normal profile. The seizures stop, and within 36 hours, we've been able to wean him off all anti-epileptic drugs, including the midazolam. He's able to be extubated, and in fact, several days later, he's discharged home. Now, the thing I want to point out about Maverick is that he had been given a trial dose of pyridoxine when he was admitted to see whether he had this condition, and he didn't respond to it. And so this is truly a case where had Maverick not received a rapid genome, he almost certainly would have died that week, and certainly he would have had permanent brain damage. And instead, he is thriving. And this is a picture of him about a month ago here in San Diego, and you can see that he's a vigorous little boy, and his mom and dad love him very much. So that's really why we are so excited about rapid genome sequencing in a NICU and a PICU. It can avoid morbidity in babies such as Maverick, and it can even prevent mortality. So let's back off now and look at the big picture. That's one case, but is this generalizable? Is this something that could be recapitulated in neonatal and pediatric intensive care units around North America? So one thing that we've undertaken in the last year is to do a meta-analysis of the entire medical literature, looking at genetic diagnosis of genetic diseases in children, either by whole genome sequencing, or WGS, exome sequencing, which is about 1% of the genome, but just about all of the genes in the genome, and then the standard test, the sort of gold standard currently, which is called chromosomal microarray. What you can see in this meta-analysis is that firstly, there are a lot of studies, in particular a lot of studies of exome sequencing, not so many yet of genome sequencing. It's just starting to be broadly used. And then there are a lot that have been performed on chromosomal microarray. And you can see the total number of subjects across all of these studies in bold at the bottom of each section. The diagnostic utility is shown over on the right, and we have the averages and the confidence intervals. So the average diagnostic rate with genome sequencing is 41%. The average diagnostic rate with exome sequencing is 36%. The average with the gold standard is only 10%. So clearly exome and genome sequencing are superior to chromosomal microarray in giving a diagnosis of a genetic disease to a child across a great number of studies. But the other thing that you'll see is that there are very wide error bars. So across the studies reported to date, there's a great deal of difference from site to site. Let's look at this in a bit more detail and understand what it means. We're going to just look at a series of studies that my group has done over the last 5 years. So the first study was actually the first 35 babies to receive rapid genome sequencing in the world, right after we invented this technology. And this was published in 2015. The study was done in Kansas City, and we selected the first 35 babies that we thought might benefit from getting their genome sequenced as fast as possible. And here are the results from that manuscript. So 35 babies had rapid genome sequencing, and in addition, because this was an experimental test, they also received all of the standard tests. The standard tests gave only 2 diagnoses, and neither of those changed how the baby was treated. In contrast, 20 babies got a diagnosis by genome sequencing, so 57% versus 6%. That was a significant difference. Second of all, 11 of the 20 babies had changes in their management, and 4 of those, or 11%, had a favorable change in outcome, and 1, their life was saved, or 3%. So a story similar to that of Maverick. So that was our very first experience with this, which was really mind-altering for all of us who were involved. We realized, wow, this is going to transform how babies are cared for in a NICU and a PICU. But again, this was the first study. It was only a small number of children. We need much more evidence if this is to become normal. So our second study was a randomized controlled trial, which is the highest quality of evidence. It was funded by NIH through a program called Insight. This picture shows the actual study design. So we assessed 129 babies to see if they might be eligible for the study. 65 were indeed enrolled and were randomized either to receive rapid genome sequencing and all standard tests, or to receive standard tests alone. So 32 got genome sequencing and standard tests, 33 received standard tests alone. Now, one thing that we noticed was that our neonatologists already felt strongly that genome sequencing was valuable in these patients. And so in many of the patients, they asked us if they could cross the baby over to receive genome sequencing. And in fact, this led to us terminating this study early. We'd intended to enroll hundreds of babies to really get definitive proof that genome sequencing was superior. We had to terminate the study early because so many of the control babies were getting genome tests. Here's the main result, the main conclusion of that study. On one axis, you can see here the time to diagnosis from 0 to 480 days. And on the other axis, the proportion of babies who received a diagnosis. And what you can see is that genome sequencing gives a higher rate of diagnosis than standard testing and a much more rapid diagnosis. And in fact, between the asterisks, whole genome sequencing was statistically better than standard testing. So this is a more rigorous test of the diagnostic benefits of genome sequencing. The next thing we wanted to look at was clinical usefulness. So yes, in this study, the Insight 1 study, we'd shown that the test was, from a diagnostic test standpoint, better than standard tests. But that didn't speak to whether it improved outcomes. And so then came our third study, which was published in April of this year, which actually looked at morbidity and mortality and cost of hospitalization. So since the prior study, I had moved with my group to Rady Children's Hospital in San Diego. And this study reported the first 42 babies tested in Rady Children's Hospital in our NICU, PICU, and cardiovascular intensive care unit. One big difference between this study and the previous studies is that we were now testing a much broader swathe of children rather than just those who were cherry-picked as having a high likelihood of a genetic disease. So let's look at the results together. So 42 babies were enrolled. and they received both standard care and rapid genome sequencing. So this is a cohort study. The standard tests diagnosed the genetic disease in 4 of 42 babies, or 10%, and one of those babies, or 2% of the 42, had a change in treatment, but it didn't change the baby's outcome. In contrast, rapid genome sequencing made a diagnosis in 18 of the babies, and 13 of the 18 had a change in management. 11, or 26% of the total, had a change in outcome. That is remarkable. We can change the outcome in about a quarter of the babies in an intensive care unit who meet these very simple enrollment criteria shown at the top. So with this study, we looked in a little bit more detail then at the economic impact. It's going to take me a little minute to unpack this slide because it's a little complex, but let's get into it. So the first baby that we modeled has Niemann-Pick type C, or MPC1, and we reckoned that the impact of rapid genome sequencing was to shorten the time to diagnosis from 15 days of hospital stay to 8 days, or a 7-day decrease. And that's because during his first admission, he had not had available rapid genome sequencing. It wasn't until his second admission that we were able to offer the test, and we made a diagnosis. And our Delphi panel argued that had the test been available during his first admission, we would have made a diagnosis earlier. And so the avoided cost was figured to be the cost of the second admission, which was $27,000. Now, he then started on an experimental treatment for Niemann-Pick type C called miglustat at 2 months of age, and we believe that he's one of the youngest children ever to have started treatment with that medication. The second child is really quite tragic. This is a baby who had been in the NICU for 250 days by the time we started to do rapid genome sequencing. He was not expected to have a genetic diagnosis, but when we tested him, we found that he had ARID1B variant that was pathogenic, giving him a diagnosis of Coffin-Siris syndrome. And given his total medical picture, his parents decided that he ought to receive palliative care. I don't have time to go into his medical condition, but it was incredibly severe. He'd had multiple surgeries, multiple episodes of ECMO, and he was in septic shock at the time his diagnosis was made, and he died the day that palliative care was started. Had he not had this diagnosis, he would have started that day treatment for this newly diagnosed septic shock, which would have lasted at least 6 weeks with intravenous antibiotics in an intensive care unit. And so we avoided, in his case, $327,000 of needless care, which wouldn't have benefited him. Now, in fact, you could argue that had our test been available early in his course, he probably would have had this test performed before his first surgery, and we may have actually saved much of the $1.9 million. But our Delphi panel thought that we could not really predict what his parents' decision-making would have been, and so they went with this more conservative number. The third case, very simple, a floppy baby, something we see every week, and this baby got a prompt diagnosis of Mendelian myopathy and avoided the need for a muscle biopsy and an electromyogram, with the muscle biopsy under general anesthesia, saving about $10,000. A remarkable case is the next one. This is a baby with Alagille syndrome. This baby was in the operating room about to undergo an operation called a Kasai hepatoportoenterostomy for treatment of a different condition called biliary atresia. And so his entire workup pointed to biliary atresia, but in fact that wasn't the correct diagnosis, and we just managed to stop that surgery from happening in the operating room. Had he had that surgery, there's a 80% likelihood he would have died. So we probably saved his life, and a 100% likelihood that he would have needed a subsequent liver transplant. And so in his case, from an economic standpoint, we saved the cost of the Kasai operation and a risk-adjusted percentage of the cost of a liver transplant. And I'm not sure what happened to my slides. I think I lost the bottom of this slide. So there are two more cases. Nothing ever goes according to plan. There are two more cases. One is a miracle baby who had a presentation identical to that of Maverick, a baby with the same EEG profile, who turned out to have a very different condition called Ohtahara syndrome. We were able to make a diagnosis in her, she's called Sebastiana, and get her on the correct treatment within a couple of days. And again, we avoided about a month of prolonged duration in the NICU because of that. She was able to go home before Christmas. And the last baby, a baby with neonatal hypoglycemia, also in that bit that's cut off at the bottom of the slide. And again, we were able to shorten the hospital stay by about a month by identifying the cause of that baby's hyperinsulinism as focal congenital hyperinsulinism, which changed the surgical procedure and allowed that baby to go to surgery a month earlier than would be normal. If we add up all of these cases and the costs avoided, it came to $800,000. And even net of testing all 42 families by genome sequencing, that was a significant amount of cost savings for the hospital. So not only does genome sequencing save lives and avoid morbidity, but at least in this set of 42 babies, it resulted in lower healthcare costs. This is something we're phenomenally excited about because it means that it's likely that this will become reimbursed and therefore something that many hospitals will be able to adopt in the next year or two. And here is Sebastiana. That's the little baby who has Ohtahara syndrome. And here's her mom and dad talking about their experience at a conference we recently had. And if you'd like to, you can read all about her story because it was in Time magazine in September 2017. And you can read all about Sebastiana in more detail. So we're coming to the end. Oh, here it is. It was cut off on the previous slide. So my slides are just mixed up. So this is the story where we were able to save $181,000 in Sebastiana's case and $125,000 in the case of a baby with hypoglycemia associated with focal hyperinsulinism. So our strategy is to bring genomic medicine first for the sickest 2.5% of children. So we've looked over claims codes in a very large database of the company Milliman of 40 million covered lives, and we figured out that about 15% of NICU admissions would meet eligibility according to their codes in terms of billing for rapid genome sequencing. And we believe that we would be able to improve their outcomes to give additional quality adjusted life years. And we looked at their charges, and each of these babies on average cost the healthcare system about $250,000 in the first year of life. So we really do believe that what I've shown you is something that's going to be generalizable. Right now we're doing a new clinical trial. We've now enrolled, actually this is a little outdated, about 143 babies in this trial, where we're comparing genome and exome sequencing and evaluating the need for either singleton testing versus testing mom, dad, and baby. And over the next year, we should have the results of this study as we enroll 250 infants. We think this will be the pivotal study to really convince folk that genome sequencing is the new gold standard for such babies. But I want to show you just one or two early results from that ongoing study. So first of all, we're screening 44% of our NICU admissions, so a very broad group of babies for this trial. Overall, we're finding that even looking at this many babies, almost half of the NICU babies, our rate of diagnosis is still quite high at 21%. And so far, we're finding that you don't need to sequence mum and dad as well, that only gains us 1% more diagnoses. What we've been asking the physicians caring for these babies is, in your opinion, is rapid genome sequencing or exome sequencing useful? 91% say yes, where their baby got a diagnosis. But more interestingly, 71% of babies who didn't get a diagnosis are still saying that genome sequencing is very valuable in helping them to manage the baby. So ruling out genetic conditions in some cases is as important as ruling in a genetic condition in terms of helping decision making in the NICU and PICU. Then we asked parents, do you feel you understand your results that we gave you? 70% said yes, 30% said sort of, but none said no. So that's very good. One of our concerns was that this might be overwhelming for parents of children in an intensive care unit. Turns out that doesn't seem to be the case. And then we asked the parents, did you think that genome testing was useful in your baby? 100% whose babies received a diagnosis said yes. 94 who received a genome sequencing didn't diagnose a genetic disease said yes, and only 4% said no. That's very surprising for us. That's much more positive than we had anticipated. And in fact, one of our concerns and the reason why we did this study was that we were concerned that we might provoke anxiety and feelings of guilt or other potentially harmful outcomes in doing genome testing in young babies. That doesn't seem to be the case. So let's wrap it up with the last slide, or the penultimate slide. Rapid early genomic sequencing in the NICU, PICU, and CVICU we feel is going to be applicable to about 10% of infants. The diagnosis rate is going to vary based on how many infants you test, between 20% and 60%. Overall, the studies are consistently showing that 31% of babies who are tested will have changes in treatment as a result of testing. Somewhere between an 11 and 26%. That's pretty broad still, but that's 10% to a quarter, 1 in 10 to 1 in 4. It's going to change the outcome for the baby in a favorable manner, and 1 to 3% is what we can expect in terms of lives saved. This is a revolution. And then in addition to that, the perceptions both of pediatricians and children's parents are overwhelmingly that this is useful in their opinion. And so this is our figure here of what we think a NICU or a PICU stay will look like in maybe 5 years' time. That these things that I've shown you today will be normal, that we will have daily artificial intelligence scrutinizing the medical record to pull out for computers to analyze what's going on in the baby's symptoms. That there'll be same-day testing looking at the genome assisted by artificial intelligence feeding off the medical record to give rapid results, and that precision medicine will be normal in intensive care units, and that it won't just be for standard treatments either, that we'll start increasingly to have clinical trials of new treatments for babies whose conditions currently are untreated, with the goal of really changing the outcomes. So that's my slides. Thank you very much for listening to me. I'd like to acknowledge that this work was done by a really big team. I'm speaking, but this took a team of about 50 people to do this work, and our collaborators are listed in the bottom right, it's taken the help of several biotechnology companies, and we're very grateful for funding support from the National Institutes of Health. Thank you very much.
Thank you, Dr. Kingsmore, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on the screen and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, what are the benefits of whole genome sequencing versus whole exome sequencing?
Boy, that's a good question. I think we alluded to this, that this latest clinical study we're in the middle of is set up to answer that question. But in the meta-analysis I showed, they appear to be fairly comparable. But exome sequencing is less expensive than genome sequencing. It's a bit slower than genome sequencing, by about a day to 2 days. So right now we're trying to weigh up the benefits versus the increased cost and decide which should become the first line of test. And right now I can't answer that, but this new study that we are starting or are in the middle of hopefully will answer that question unequivocally.
Our next question is, what needs to happen in order for the whole genome sequencing becoming a part of standard care for babies with rare and undiagnosed diseases?
Well, that's a tough question. 3 things need to happen. First of all, what I'm showing you is still somewhat experimental. We're a research institute, and so it's not available in most children's hospitals around the country and around the world. And so the first thing we need to do is what we call implementation science, which really means demystifying genome sequencing and making it available to children's hospitals worldwide. We're in the process of doing that, as are others. And by the end of this year, we predict that this will be available in probably about 75 children's hospitals around the world. So that's the first big step. The second big step is to build the evidence base. That I today reviewed the world's evidence base, and really it's only 4 studies. There might be 1 or 2 others that you would say as part of the evidence base, but it's certainly less than 10 studies. So as a community, a scientific community, we need to build the evidence for when this works, how it works, and persuade our colleagues all around the world that this is the best treatment and when it's the best treatment. And then the third thing is actually to educate intensivists, that's neonatologists and doctors in intensive care units. They're not used to this. They've probably never used this. They're not familiar with these principles that I shared with you today. And so there's a big job to do to educate them. I said there were 3, but there's actually 4. The 4th one is that right now, this is still an experimental test. It's not reimbursed by payors, and that's another piece of the puzzle. This needs to be covered by your health insurance plan so that you can be able to get this without paying out of pocket.
Dr. Kingsmore, how is RCIGM working to expand access to rapid whole genome sequencing?
So as we develop these results and we're flabbergasted by them, we said, you know, we really have a mission to share this with the world. And so over the last 2 years, we've been on that odyssey of switching on one hospital by one hospital by one hospital. The first place we switched on was Minnesota Children's in St. Paul and Minneapolis. And the second was Colorado Children's in Denver. The third was Children's Hospital of Orange County. And now today we're live in 6 or 7 children's hospitals. And as I say, we're growing that number. And by the end of the year, hopefully, we'll be in about 75 children's hospitals. We would like over the next 5 to 10 years this to be offered in every children's hospital in the world as part of the standard of care.
And it looks like we have time for one more question. How do you expect the field of genomics to evolve in the next few years? And what can healthcare organizations do to prepare for this change?
Yeah, that really ties into that first question on my last slide. What does the NICU of the future look like? And I think that omics testing genomes are going to be pretty normal. We're going to have to train our physicians so they're used to that. They can read that, they can speak it, they can speak genome. Our entire hospital systems need to be retrained. Our nurses, our genetic counselors. So what can people do to prepare? They can get involved. Watching this video is a good start. Then the next thing would be to reach out to us. We are going in the next month or two to start to have monthly live WebEx-based case conferences. They're intended for physicians all over the world to just plug in once a month and hear a story like Sebastiana's or Maverick's, so that together we can learn from one another in this great new way of managing the sickest of babies in our hospitals.
I would like to once again thank Dr. Kingsmore for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I would like to let everyone know that today's webcast will be available for on-demand viewing through September of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.